Welcome to my presentation about the standardization of extracellular vesicle concentration measurements and update on METVES II. So first of all, I have no relevant conflicts of interest to declare. Extracellular vesicles are nano-sized particles, which were formerly also called microparticles, and they can be secreted by all types of cells and can be found in all types of body fluids, such as, for example, urine or plasma. And the idea is that the EV concentration might serve as a biomarker for diseases such as cancer or cardiovascular disease. And how that could look like is that you're collecting blood from a patient, for example, as you can see on my slide here. To the right, you're collecting the blood, you're removing the cells, and then afterwards, you're measuring the EV concentration within your sample. And then you can compare the EV concentration within your patient cohort, but you could also compare it between your patient cohort and healthy individuals. And one way to detect vesicles is by flow cytometry. And I just showed this in a very schematic way here quite quickly. So how that would work is that you're having your particles in a flow, they're illuminated by a laser beam, and then the fluorescent intensity as well as the scatter intensity in arbitrary units are measured. And over the past couple of years, awareness has actually grown that the EV concentration measurements are incomparable between different instruments. And a lot of standardization studies also have been conducted over the past couple of years, but I'm not going into too much detail here about them because I think it's rather important to focus on the lessons we've learned from them, which are, for one, for example, that the instruments differ in sensitivity, and also that most of these studies were actually performed on uncalibrated flow cytometers. And what that actually means, I'm going to explain now. In general, EV flow cytometry is difficult. And why is that? As I said, for one, the instruments differ in sensitivity, and I just brought a little example to explain that here. So if we are looking at the concentration of vesicles versus the diameter, and we are assuming we measured the same sample on different flow cytometers with different limits of detection, we might get the following out. So if the one flow cytometer had a limit of detection of around 150 nanometer, so that's shown here with the green line, while the other one would have a limit of detection of about 500 nanometers, shown here with this blue line, we would end up measuring a 30-fold difference in concentration between these two instruments, even though when measuring the same sample. So a kind of easy trick to circumvent that is that you compare your data within the same size ranges of EVs here. Another thing is that EV flow cytometry is difficult because the data comes in arbitrary units, which makes it impossible to interpret the data for one. And the other thing is that it makes it impossible to compare your data. And consequently, at the moment, we can't conduct any multi-center studies because of this. And that's actually a problem because if we want to explore EVs as biomarkers, we need reliable measurements, but we also need comparable measurements. And one thing to solve this problem is to calibrate your instrument. So that means we're relating the arbitrary units that you measured, which don't really tell you much actually, to comparable standardized units. And that, for one, enables you to do data interpretation properly and also to do data comparison. But a calibration requires reference materials. And at the moment, we're unfortunately facing a lack of well-characterized reference materials for EV flow cytometry. But I'm going to come back to that. I just brought a little example here of one sample, that EV-containing sample, of course, that we measured on two different flow cytometers. And so we measured it on an Apogee 60 micro at our lab in Amsterdam, and Joanne Lanigan measured it on a Cytec Aurora in the US. So what we can see here is the fluorescence in arbitrary units versus the side scatter in arbitrary units. And one of the things here actually is that we are not even sure if we look, so within this gate that we set here, so with the particles we're looking at, if they're even in the size range of EVs or if they're much larger. So what we did is we calibrated the instrument, or we calibrate the data as well. And then we get standardized units out of it. So for example, we are looking at the molecules of equivalent soluble fluorochrome now. So that would be the fluorescent intensity and the diameter in nanometers. And now we finally can compare the concentration of vesicles between both instruments. So what does this actually tell us? Yes, calibration works. And if you want to have comparable data and want to compare it to another lab, please perform a calibration. And one project that actually aims to achieve comparable EV concentration measurements is the METVES II project. And what we are aiming to do here is we want to develop one reference material to calibrate them all. What does this mean? This means we want to have one reference material that calibrates all aspects of a flow cytometer, such as the flow rate, the fluorescent intensity, and the scatter intensity. And we want that, or the developed reference material has physical properties that are comparable to EVs if it comes to the number concentration, if it comes to the scatter pattern, and also if it comes to the fluorescent intensities. And at the moment, we are developing 3 different types of reference material at the project, which are hollow organosilica beads, polymer-based solid low refractive index particles, and also liposomes. Another thing we are developing within the project is a quality control sample, which is a ready-to-use EV test sample that will be later on used to validate the newly developed reference materials. And we're developing this test sample from either human urine or human plasma as a starting material. The sample contains pre-labeled EVs and is stable over time. So if we're looking at the concentration versus time in months of storage, we can see that it's pretty stable over time. And one of the things we're also doing within the project is that we're conducting an interlaboratory comparison study, which we're organizing at the moment. And this is to, first of all, validate the METVES II reference materials, which are supposed to calibrate all aspects of a flow cytometer. And then after, we want to measure a comparable EV concentration in this ready-to-use EV test sample. And the study will actually start in October of this year. And that brings me already to the take-home message. So first of all, I think it's very important, and I hope I brought that across properly, is that if you want to explore and exploit EVs as potential biomarkers, we need reliable measurements, and we need to be able to compare our data with other labs. And the METVES project aims to achieve exactly that. So to measure comparable EV concentrations by developing one reference material to calibrate them all, and by developing a ready-to-use EV test sample from different body fluids. And this will actually be the starting point for multi-center studies, which we really need if we want to bring EVs to the clinics on the long run. So with this, I'm coming to the acknowledgements. I would like to thank all my colleagues from the Amsterdam UMC and also my partners at the METVES project, obviously, and the funding agency. So thank you very much for your attention. And if you got any questions, please feel free to ask.